Okay na? I'll give you 10 minutes to write your whole timeline. Starts now. Yes.
Mali yung
Yeah.
MH also.
Yung mga dates lang dito. Tapos yung tao na walang dates, pasulat na lang yung contribution nila. Separate sa timeline, although part sila ng timeline. Any format lang.
Kaya ba? Wait lang. Ulitin ko.
Oh. Dapat yun isasama. Isasama mo.
Raise your hand.
Lahat sila. Kita lang nila. Tutunatin.
Hindi ka tapos sa level.
Kasi yung kay Griffith parang Frederick eh.
Yung wrong spelling yung ata yung mga nasa bar. Yung nasa bar with friend. Pero yung sa right
Two ways to spell his name.
Okay. Kala ko. M-I-I.
S-C-H-E-R.
Yes. Yeah.
M.H. Lawson.
Ano po yung pangalan?
E-A-W-S.
Wala siyang
J-L-I-N.
Wala bang dates si Marisa.
Oh my God. Galing. Latest version pa yan.
So dito na lang. Kasi ang nangyari sa 1930, dito na-discover yung yung
Yeah, also B. Avery.
Yung nasa PowerPoint, yung letter T.
And just say the landmark experiment.
Call it name.
Kahit landmark experiment na lang nila. Or yung contribution. Landmark experiment and contribution. It has to be complete. Date, name, contribution. Or slash landmark experiment.
Oswald. Nasa PowerPoint kasi may letter T. Oswald. Lagyan mo na lang ng letter T. Tapos si Colin. M. Dito.
Sa isang year lang.
Magkasama sila ang usap. Isa lang. May MAC
Sorry,
Yung sa McCarthy ata yung
Macleod.
alam ko din si Macleod ay MAC.
Macleod. Si McCarthy MC.
Capital.
Yung
McLean McCarthy, MACLIN.
6 minutes.
Ayan.
Martha Chase. Kasi Alfred Hershey.
Ayaw. Good. Ano ulit?
Fio.
Alin? Ay, okay. So, sino may ano? May record and thank you. Sobrang pangit yung sulat ko ngayon kasi bagong busy na ako. Ito yung tatlo. Yung first visitor na yun natin. Alin? Pati na yun yan. Pero hindi na kasama.
Basta alam ko sa
Iba yun. Ako kapatid.
isa pa siya kasama eh. MAU.
MAU. Tama. Iba yung naisip ko ng Morris. RIC. Yun ah.
Pero pwede nyo nang i-sulat yung, like yung wording para sulat. Kasi,
Martha Chase the blender.
hindi yung ano na lang, yung tatlo mo lang din. Si Aloy, si Discipline, si Calvin Collins.
Sa kanila,
Oo, siya. Yung, yung S35, tsaka
yung art is stream. Silang dalawa.
Individual daw sila. Tapos meron pa yung chemical nature of
magkasama sila ng Rosalie. Siya yung nanguna sa
2 minutes.
tapos yung base pairing yung sa kanila.
Sina Erwin.
Sharda. Erwin Sharda. Fister.
Equal yung AT plus AG.
Ako na, sige. Ay, saan ka siya? Okay na yan. Okay pa. Sa likod na. Yung walang ano. Ay, sorry. Yung, wait lang. Wait lang.
Oh, may naalala.
Si David.
Sige.
Ito, sulat niyo na to.
Ito, ano?
Okay, 1 minute.
Ilog ka din.
Chemical of drugs. Parang substance.
Alright?
Ma'am, 5 minutes.
Anong 5 minutes? Super. O sige, 10 seconds.
Ang alam ko parang
Finalize your answers. 5, 10, 15, 20, 25, 30. 40 points.
Sa past.
40 points siya, pero re-ratio din naman siya. It's just a component. Pero it's crucial.
Discovered isolated.
A point can
First
change your life. Can change the trajectory of your life.
Use times plus isolated.
Alright.
Tama, isolated. Isolated. Use time.
Okay, wrap it up. Okay na?
30 seconds, ma'am.
Anong 30 seconds?
Ma'am, 30 seconds, ma'am.
Okay. Pakisulat ng names nyo. Nasulat na ba?
Sino may
Last name na lang. Pakisulat last name na lang. Huwag nyo na po para mabilis. Okay, please pass your papers. Pass your paper to Wala.
Pwede na po.
Pass. Pass your paper.
Sa likod yata, may parang ito rin yung
Hatch keys yung pangalan niya. Okay lang yan. Hatch keys.
Alright, huwag mo na kayo bumalik sa proper seat nyo. Okay. Please, change paper muna kayo. Ayan. Mag-check. Mag-check mo ito. Please check. Taray, di ba? Okay. Alright, so while we are checking, so magdi-discuss tayo, and then as we go through discussion, nag-check kayo ng work ng other groups. Okay? So the first, pinaka-earliest here is, pinaka-earliest pa? Earliest here is 1869. So ano nangyari noong 1869? Sino yung key scientist in 1869? So that's Friedrich Mischer, or kung yung spelling ko Friedrich, okay din naman. You can spell his name two ways. So, sorry, if German yung person here or French, baka ma-murder ko yung name niya. So, sorry na lang in advance. So, ano yung contribution ni Friedrich Mischer during 1869? So, this scientist is a Swiss physician and biologist. So, he's the one that isolated this substance called nuclein. Okay? So, this is the chemical of nucleus, and it is known to be acidic, phosphorus-containing long molecule. And this one, he isolated this from soiled bandages or fossils in bloody bandages. So, sa ano nyo, dun sa nag-check, please 5 points pag kumpleto yung pangalan, the date, and its landmark experiment or landmark discovery. Okay, 5 points. Pag kulang ng isa, kasi for example, mali yung landmark discovery niya or landmark experiment, just 3 points. Pag wala siyang sample date, tapos mali yung pangalan, 0 na yon. Okay? Pero kung may date at tama yung pangalan, that's 3. Pero pag meron siyang contribution, that's 5. Pag mali yung date and date, that's 0. Yes.
By maling name, you mean ma'am, kasama din yung spell.
Oh, good question.
Di ba
Yes.
may
Kasama ang spelling. Pwede ko ma-murder yung pronunciation pero hindi yung spelling. Pero two ways naman yung pag-spell ng pangalan niya. Fried, F-R-I-E-D. Frederick, paano yung pronounce nun? R-I-C-H. Frederick, it's the same pronunciation.
It's the same pronunciation.
Same pronunciation. Okay. Why do we have certain arguments now back then that DNA is not the genetic material? So arguments against DNA as the genetic material because first, it is too simple. And they know that yung proteins are more diverse than DNA. Aside from that, DNA is only relevant to some bacteria, but protein is more likely. So protein, they think, is more likely the genetic material. And aside from that, the extract that was isolated might be contaminated. Hence, it could not be a pure DNA. So still, it can be associated by other biomolecules. So maybe they think DNA is not the genetic material. Now, let's take a look at the timeline. Again, yung discovery or different landmark experiments that brought about DNA that is established as the genetic material. So after 1869, the next is 1928. So we have here Frederick Griffith, not Frederick. So Frederick Griffith, 1928. So ano yung kanyang landmark experiment? It is the transformation experiment. Okay? It's the transformation experiment on Diplococcus pneumoniae. Okay, so it is a bacteria. So your Griffith experiment, meron siyang dalawang pinag-aaralan na strain. Two types of strain. So the first type is the smooth type, and the other one is the rough type. So the smooth type, it contains this thick polysaccharide capsule. So it's smooth and mucoid colonies yung itsura ng bacteria. And then, it is known to be pathogenic and virulent. However, the rough type 1 has no capsule and then it contains rough colonies and non-pathogenic or avirulent. Why do you think na considered as avirulent yung rough strain na bacteria? Kasi walang capsule. Bakit? So, ano yung significance ng capsule? Yes.
Yung capsule ko yung nagpo-protect sa immune system ng hosts. So, technically,
Ng bacteria. Kasi if wala siyang capsule, the immune system ng host, ng tao, for example, can attack the R strain kasi wala siyang protective coat. Okay? So, yung R strain has no coat because it can be attacked by the immune system of the infected animal. Okay? So, that's why. So, let's take a look at the brief summary of Griffith's experiment. So, we can see here, may gumamit siya ng rough strain. And then, this rough strain is known to be non-virulent. And then, it was injected to a mice or mouse. So, do you think the mouse live or die? So, it lives. Nakikita nyo naman sa illustration. Buhay siya. Okay, so it lives since it's a virulent and it's not pathogenic. Now, the smooth strain, known to be virulent or pathogenic, inject it to the mouse, so you expect it to die. So, namatay yung mouse. Now, the next thing ang ginawa, this smooth strain, ito that pathogenic strain was heat killed. So once it's heat killed, it was injected to the mouse and still, of course, the mouse lives. However, there is this last experiment ang ginawa. The heat-killed smooth strain was mixed with the rough strain bacteria. So what do you expect? Supposedly, di ba dapat mamamatay siya? Kasi na heat-killed naman yung smooth strain. Kasi from the previous experiment, the mouse lives. So hindi namatay yung mouse. However, nung dinagdagan siya ng rough strain with the smooth strain na heat-killed, it still died. The mouse died. So what might be the reason why? Let's take a look at this. So, the live S or the live smooth strain, the mice na discover niya is S strain na bacteria and the mice is dead. For the live R, the mouse is alive, she's a virulent to, so none of the S cells was discovered. For the heat-killed S, the mice is alive, and none of the S strain cells were recovered. Now, for the heat-killed S and the live R strain, the mouse was dead, and then, surprisingly, they discovered the S strain bacteria. Okay? So, therefore, there is this transforming principle that resurrects, that R-types was resurrected the S-type. So, yung R-type daw, with the use of R-type, parang na-resurrect yung S-type. So, there is what we call transforming principle. Good morning. Kanino po ito? Patayin ko yun. Okay. So, therefore, information daw from this heat-killed S strain was transferred to the live R-type that transformed it to become virulent or pathogenic. Okay? So, this experiment of Griffith is actually, kumaga, controversial at that time, but many people did not believe him. Aside from that, Griffith is known to be a very shy scientist, and he cannot communicate his work very well. So, meron siyang problem doon. So, imagine if you present to a lot of people and you are very tense, hindi mo alam yung sasabihin. Alam niya, he has a good result, however, it was not communicated very well. Hence, it created such skepticism in the scientific community. Now, ang ginawa after that, so after Griffith's experiment, he actually died after World War II because he refused to leave his lab during air raid. So, RIP. Okay, so the next experiment was in vitro transformation. So that is by M.H. Dawson and J.L. Alloway. So that is around 1930 and 1932. Tama ba yung ano nyo? Yung kaklase? So this landmark experiment was in vitro transformation. When we say in vitro, this one is an experiment that does not need that mice anymore. So they did was precipitation of transforming substance. How did they do that? They have this heat-killed S, the S strain, plus the live R extract, which contained the live S strain. So ito yung previous na experiment ni Griffith. So in-incorporate ito so the following year by Dawson and Alloway. So let's take a look at this. So this is the general procedure of how he did or how they did in vitro transformation. So the first one here, you have your S strain that is grown in culture so that you can have enough cells to be studied. So, ayun. And then after that, this is there is lysis and precipitation. So you can get the cell-free extract. When you say lysis and precipitation, the S cells are killed or lysed so that yung internal components niya, all of that will be released. So wala ka ng, so it will be a cell-free extract that will produce all cellular components, including that transforming substance. Actually, we know that as DNA right now, or DNA fragments. And then after that, this cell-free extract was mixed with our strain culture. So the same thing that happened in Griffith experiment, but this one is in vitro. Now, this R strain culture plus this cell-free extract, so this was mixed. And then after that, nagkaroon ng tinatawag na transformation. We actually have transformation talaga na methods in molecular biology. So transformation of some of the S cells. So nagkaroon ng S cells from that live R strain. Okay? So in this number 4 na, sorry, wait lang po Hindi ko siya nakikita. Okay, so number 4, you can see here. So this is the actual process that happened. So this one is your S cell. It was heat killed. So pag na-heat killed siya, this will be lysed. And then we have here DNA fragments. So this one is, let's assume this is the one that codes for that smooth capsule. Okay, that makes that particular strain pathogenic or virulent. Now, what happened is these S cell destroyed DNA fragments remain intact. So it's still intact. And then this one was mixed with the live R strain. So ang nangyari, there's what we call transformation. This live R strain, inuptake niya or tinake in niya yung fragment or that DNA fragment that contains the S gene that codes for that smooth capsule. Hello? Now, with that, meron ka ng transformation ng live R cell into that virulent S cell. You follow? So basically, tinake in niya yung DNA fragment. Kaya after the experiment, some has the still live R cells, but the others may na-retrieve na live S cells. Follow? So that is basically the transformation. So in conclusion, the transforming principle or agent is responsible for the transformation of the avirulent R type to the virulent S type strain. So that is the conclusion of Dawson and Alloway. The next one is during 1944 rather, sorry. Tagalog na 1984. So we have Oswald Avery, Colin MacLeod, and Maclean McCarthy. So what they did is experiment on the chemical nature of transforming substance. Okay? So basically what they did is to destroy that transforming substance or DNA para ma-prevent yung transformation. So for example, if they degraded or destroyed, not degraded, it's actually destroyed. When you say degraded, there would be still fragments of DNA. Destroyed that substance so that hindi magta-transform into live or cells. Now, let's take a look at the general isolation step. So, when we say isolation step, this is actually the genomic DNA isolation of bacteria. So, sino dito kumukuha ng 120? If you're taking 120, you will do this, but it's not bacteria though. It's more on plants. So, this is DNA isolation. So, the first one, they did bacterial culture. So, meron large-scale growth ng Streptococcus pneumoniae. so that we can obtain enough cellular material for the experiment. And then next is centrifugation. We need to separate the bacterial pellet from the growth medium. Kaya kailangan natin ng centrifugation. Number 3 is resuspension of that pellet in 150 ml chilled NaCl. So that is to maintain the osmotic balance that will stabilize your genomic DNA. Okay? It stabilizes genomic DNA. And then the next one is heat. Heat that kills 65 degrees. Heat kills at 65 degrees Celsius and wash with saline solution. So it's actually basically lysing the cell up, opening the cells to release DNA. And then after that, we have your saline plus deoxycholate to extract water-soluble cell components, and that still includes DNA. And then we have ethanol precipitation that helps concentrates and precipitate your nucleic acid or DNA still. And then you have your chloroform extraction. So your chloroform extraction is actually very important to remove other biomolecules such as lipids or proteins, at yung matitira na lang is yung DNA or that nucleic acid. And then finally, you have your enzyme digestion to eliminate polysaccharides so they do not interfere with transformation. And then ethanol precipitation that will further purify and concentrate your DNA. Okay? Now let's take a look at the general properties of the transforming substance. The first one is it has to be viscous and silky sheen. So, sino dito yung naka-experience na mag-isolate na crude DNA ng strawberry? Or any crude DNA ng plant? So, di ba, after that, you can see a very whitish substance. So, that is actually one of the general property of the transforming substance, which we know right now is DNA. The other one is it loses activity in distilled water because your DNA requires, of course, ions to maintain its stability, if you know the properties of DNA. Then, activity now is remained or retained for months at physiological salt solution. So, your salt actually stabilizes DNA. And then it can withstand heating at 65 degrees Celsius for 30 to 60 minutes. So, yan yung mataas yung heat stability ng nucleic acid, particularly DNA compared to proteins. And then, it loses activity with increased acid concentration. Okay? Now, let's take a look at the qualitative chemical tests that they did to identify the chemical nature of the transforming substance. Now, for protein, they start using Biuret or Million test, and it's negative. They also did the lipid chemical test using chloroform ethanol extraction. It's still negative. They did for RNA, orsinol, it's weakly positive because we know that some of the nitrogenous bases in RNA is also present in DNA. Then for the DNA, they also have this experiment, the dish diphenylamine experiment, and it's positive. So therefore, they can conclude, maybe this is really DNA and not protein as the transforming substance or the genetic material. So other elementary chemical test, yung kanyang nitrogen phosphate ratio, it's around 1.67. And yung kanyang UV absorption, yung kanyang lambda max, or it can absorb light maximum at 260 nanometers. And that is the property of what we call the nucleic acids. Right? Your nucleic acids can absorb approximately the highest absorbance or the lambda max is 260 nanometers. Yung protein, di ba, is 280, tama? 280 nanometers for protein. For nucleic acids, particularly DNA and RNA, is 260 nanometers. Now, so yan yung chemical nature ng transforming substance. And for enzymatic analysis, if they added protease, still, nagkaroon ng transformation. That live R was resurrected, quote-unquote, into the live S strain. They use lipase that degrades what? Lipids. Still, the transformation happened. They also use RNAs that degrades RNA. Still, transformation happened. But when when they added DNAs that degrades DNA, there is no transformation. So hence, it is DNA that is the transforming principle. Because when they heated the DNAs, nagkaroon ng transformation. You follow? So that is how they establish DNA as the genetic material. So basically, siya yung transforming substance. Or the transforming principle, rather. So, tingnan nyo yung work ni Griffith, nakaabot dito. So, it has been, there were further experiments after Griffith that established DNA as the genetic material. So, this is just the general illustration of how they did that. So, DNAs plus R strain, it's alive. So, na-resurrect, ano siya? It's alive kasi namatay yung transforming substance. But the rest, it's all dead. Okay? Have you watched the blender experiment? Sino hindi naka-watch? Siyempre, panonoorin natin siya. Okay, later. Okay, now for 1952, we have Alfred Hershey and Martha Chase. So that's 1952. Ano yung kanilang landmark experiment? That is the blender experiment. Okay? So they have this hypothesis now that DNA is essential for the production of the new phages, while protein coats are only for attachment. Are you familiar with bacteriophages? These are viruses for bacteria. Very good. MCB. Okay, so what they used was T2 phage. So they lysed the cell after 30 to 60 minutes to release new phages. So they labeled or radioactively labeled the phages. Sulfur 35, that is for the protein, and you have phosphorus 32 for the DNA. So your protein contains sulfur, particularly what amino acid, cysteine and methionine. Tama ba? And phosphorus is basically, it contains the backbone of your DNA. So that's why it was radioactively labeled with phosphorus 32. Do not forget sulfur 35 and phosphorus 32. Baka nasa quiz. Okay. So these phages were labeled and protein coat with 35S and DNA with 32. So let's take a look at blender.
In 1953, Alfred Hershey and Martha Chase confirmed DNA's preeminent role in genetics by demonstrating head, sheath, tail, and base plate made of different proteins. DNA is packaged within the head of the virus. When T2 comes in contact with E. coli, the phage
Oh,
attaches
it's not
to the
moving. It's not moving. Okay siya.
head, sheath, tail, and base plate made of different proteins. DNA is packaged within the head of the virus. When T2 comes in contact with E. coli, the phage attaches to the bacterium by its tail. Next, the phage injects genetic material into the cell. The genetic material directs bacterial enzymes to produce viral offspring. When the life cycle is complete, 100 to 200 progeny phages have been assembled inside each bacterium. The bacterium breaks open, or lyses, and the phages are released. Since T2 consists of only DNA and protein, Hershey and Chase reasoned that the genetic material must be one of the two components, so they designed an experiment to determine which it is. For their experiment, The scientists prepared T2 phages that either had radioactive DNA or radioactive proteins. To make the components radioactive, Hershey and Chase infected E. coli with T2 and grew the bacterial cells in two different culture media, one containing the radioactive isotope phosphorus-32 and the other containing the radioactive isotope sulfur-35. All the phages released from the bacteria grown in the phosphorus-32 medium had radioactively labeled DNA, while all the phages released from the bacteria grown in the sulfur-35 medium had radioactively labeled proteins. In the next phase of their experiment, Hershey and Chase infected E. coli with the two types of labeled T2. They then used a blender and a centrifuge to separate the protein parts of the phage, called the phage ghosts, from the rest of the infected cell. The scientists discovered that the labeled DNA appeared in the host cell, but not the phage ghosts, while the labeled proteins appeared in the phage ghosts, but not the host cell. In addition, the progeny of the T2 with labeled DNA were radioactive, while the progeny of the T2 with labeled proteins were not radioactive. Because genetic material is passed on from parent to offspring, Hershey and Chase concluded that DNA had to be the genetic material of T2. Their results helped convince the scientific community that DNA was the hereditary material.
Alright, so that is the blender experiment that helped further or strengthen the establishment of DNA as the genetic material. So basically, this is just the overall illustration on how did they did the experiment. So after the blender, so meron silang two setup. The one that is labeled with 32P and the other is 35S. And then after that, after centrifuge, they test it for radioactivity. So centrifuge, and then this one, yung it was separated yung kanyang cell debris, and then the phages are lighter, so nasa taas siya. So it is discovered na yung 32P, it was found in the bacteria. Kasi diba ini-inject ni T2 phage yung kanyang genetic material dun sa bacteria. So hence, that is one reason why they concluded that DNA is actually the genetic material. Alright. So, for the other crucial researchers, so we have here Phoebus Levine. So he's the one that concluded that DNA is linear. Just don't put any date on it, just the name and its contribution. And he thought that DNA is made up of nucleotides with 4 different bases. So ito yung kanyang model. So medyo, ano siya, weird. Okay. And then, actually he concluded that DNA is actually tetranucleotide. Okay? Tetranucleotide. This is actually 1929. Just so you know. 1929 to, pero hindi lang nilagyan ng date. And then we have here Erwin Chargaff, Ernest Fisher, and R. Hotchkiss. So hindi lang pala si Chargaff nag-establish ng 1:1 na purine-pyrimidine ratio. So the total number of adenine is equal to the total number of thymine, and the total number of cytosine is equal to the total number of guanine. Now, this one was around a year before, actually 1952. Around 1952 ito. And then we have Linus Pauling. This is also around 1952. So he proposed that DNA is actually triple-stranded molecule, which is actually hindi totoo kasi yung kanyang model, yung phosphorus daw is inside. You know that phosphorus is negatively charged, right? So it must not be inside, it should be outside. So medyo hindi tama yung kanyang model, and so Francis Crick and James Watson was like, okay, it's wrong, we can still do something. Okay, let's revise this. And then what happened is that we have this hulog ng langit. Rosalind Franklin. with her famous photograph. Ano tawag doon? That's this photo 51. So this photo 51 was actually the one that revealed that DNA is actually helical, and it has this constant diameter. So Rosalind Franklin provided the X-ray diffraction photograph of the crystalline DNA. Okay, so that is also she was working, not really working, but with Maurice Wilkins. But they were in the same college, which is the college, King's College London. So actually, Maurice Wilkins, the one that provided this photograph to James Watson and Francis Crick. Okay, full credits to Rosalind Franklin. And then the last one, this is in 1952. In 1953, the last. So this one is the actual discovery or establishment that DNA is double helix. It's a double helix structure. They actually built this molecular model. Ito, this is a famous photograph. They built a molecular model that integrated the experimental evidence coming basically. Yung main contribution dyan is the photo 51 of Rosalind Franklin. And then after that, they made a paper and then published it in Nature in the same year, entitled The Structure of Deoxyribonucleic Acid. Okay? And, they also incorporated the idea of Chargaff, that adenine is equal to the number of thymine, and then cytosine is equal to number of guanine. So that one purine is equal to one pyrimidine. With that idea, and then with photo 51 of Rosalind Franklin, James Watson and Francis Crick came about with this model. Okay? And then finally, let's take a look at this. Have you seen this one? So this one is the actual history of the DNA and how it was established as the genetic material. So this is the last slide.
In the early 20th century, physicists and chemists unlocked secrets of the atom that changed the world forever.
Yes, it changed the world forever.
But life remained a profound mystery. Among life's deepest secrets was inheritance. Everyone knew that traits like the shape of a pea pod or the color of eyes and hair were passed on from generation to generation. But no one knew how such information was stored or transmitted. Scientists were convinced that there had to be a biological molecule at the heart of the process, and that molecule had to have some pretty special qualities.
The three-dimensional arrangement of atoms in those molecules had to explain the stability of life, so that traits were passed faithfully from generation to generation, and also the mutability of life. You have to have change in order for evolution to happen.
The challenge of solving this mysterious arrangement of atoms, this fundamental secret of life, was taken up in 1951 by two unknown scientists. Less than 18 months later, they would make one of the great discoveries of the 20th century. They met and joined forces at the Cavendish Laboratory in Cambridge, England. One was a 23-year-old American named James Watson.
He had a crew cut when he first came to Cambridge, and that was very rare in Cambridge in those days. He liked to wear what I call gym shoes and leave the laces untied and things like that. He was quite an enfant terrible, I would say. But behind that, of course, was his extreme, intense love of science right from his early years and his determination.
The other was an Englishman named Francis Crick. Trained as a physicist, his academic career had been interrupted by the outbreak of the Second World War. It wasn't until 1949 that he got back into academic science. He was anxious to make up for lost time, and now interested in biology. Crick and Watson connected instantly when they met in 1951. They both loved to talk science.
Francis and I both liked ideas, and as long as I could talk to Francis, felt every day was worthwhile.
Crick was always ready to share his thoughts, though he rarely did so quietly.
any room he was in, he was going to make more noise than anyone else.
They would constantly throw crazy ideas at each other, dismiss them, have another idea, follow that a little further, dismiss that, but then something comes out of left field. So it's kind of this give and take.
We did have different backgrounds, but we had the same interests. We both thought that finding the structure of a gene was the key problem.
The idea of the gene dates back to Gregor Mendel's experiments with pea plants in the 1860s. By the 1920s, genes had been convincingly located inside the nucleus of cells and associated with structures called chromosomes. It was also known that chromosomes are made of proteins and a nucleic acid, deoxyribonucleic acid, or DNA. That meant that genes had to be made of either DNA or protein. But which was it? Proteins seemed the better bet. There are lots of different kinds of them, and they do lots of different stuff inside the cell. In contrast, DNA didn't seem very interesting. It's just repeated units of a sugar linked to a phosphate and any of 4 bases. The readiness to dismiss DNA was so entrenched that it persisted even after Oswald Avery showed that it can carry genetic information.
Avery had isolated a substance that conveyed a trait from one bacterium to another. And this transforming principle, as he called it, he showed that it was not destroyed by a protein digesting enzyme, but was destroyed by a DNA digesting enzyme.
Watson and Crick were among the few who found Avery's work persuasive. They thought genes were made of DNA. They also thought that solving the molecular structure of the molecule would reveal how genetic information is stored and passed on. At the time, a powerful technique for solving molecular structure was being perfected: X-ray crystallography.
At its best, X-ray crystallography can determine the position of every single atom in the molecule that you're analyzing with respect to every other single atom.
Not that it's easy. The picture you end up with is a diffraction pattern, and to make sense of it, to work out where the atoms are, involves interpreting lengthy calculations. And in the 1950s, the equipment was primitive and difficult to maintain. The X-ray sources weren't very bright. And on top of that, DNA is not an easy molecule to work with.
Basically, you picture snot. And it's kind of hard to pick it up and do stuff with it and analyze it. Polymers are not fun to work with from that point of view.
Cavendish was famous for X-ray crystallography, but the director of the lab didn't want his staff X-raying DNA. He knew that a group at King's College in London was already doing that, and he didn't want to be seen as competing.
It just wasn't good manners.
The King's College scientist who had initiated the work on DNA was Maurice Wilkins. Like Crick, he was trained as a physicist and had only recently become interested in biological questions. Though he was drawn to the problem of the gene, Wilkins lacked Watson and Crick's burning urgency to find a solution. Complicating things for Wilkins was his relationship with his colleague, Rosalind Franklin. She was a talented crystallographer, but when she joined the team at King's, she believed that she would be leading its DNA research.
She had the notion that this was her project. He had the notion it was his project, and if anything, she should help him in his effort to solve the structure. And so this is a recipe for disaster.
The times and their personalities worked against an effective partnership.
This was a time when it was very, very hard for women in science to be taken seriously. And so I would imagine that Rosalind Franklin had to be perhaps quite assertive.
She certainly asserted her independence. Wilkins, by all accounts a shy man, reluctantly agreed that they would work separately. London is only 75 miles from Cambridge. That means that Watson and Crick could easily keep tabs on the work being done at King's. But another potential competitor was thousands of miles away, in California. Linus Pauling was renowned as the greatest physical chemist of his generation. He was widely admired for his ability to build accurate models of complex molecules. Watson and Crick were convinced that it was just a matter of time before Pauling used this technique to solve DNA. Biological molecules come in a variety of shapes. Pauling, and Watson and Crick, suspected DNA might be a helix of some kind. But if so, how were the sugar, the phosphate, and the bases arranged? Early in his collaboration with Watson, Crick had worked out mathematically what the X-ray diffraction pattern of a helical molecule should look like. Shortly afterwards, Watson went to London to hear Franklin report on some of her recent work. When he got back, he told Crick what he remembered of her talk, and they decided to build a model. In a few days, they had one. It was a helix, with 3 sugar phosphate chains on the inside, and the bases sticking out.
At that time, the only interesting thing about the DNA molecule is the bases. And so it made perfect sense. I mean, only an idiot would put them inside, because then they're hidden.
They invited Wilkins and Franklin to come and take a look. Unfortunately, Watson had misremembered some of her key measurements. Franklin saw this immediately and quickly and derisively dismissed their effort. She went on to craft a mocking announcement for the death of DNA as a helix. It was an embarrassment that did not sit well with the Cavendish leadership.
We were forbidden, in a sense, to work on DNA.
The failure of the first model was painful, but it can also be seen as just part of the scientific process.
I would actually maintain that in order to arrive at the right solution, you have to put out a couple of wrong ones. And that's just the nature of discovery. And if you're afraid of making a mistake, you're going to fail in this business.
Through 1952, Watson and Crick read and talked over anything and everything that could prove relevant for their ongoing, but now underground, quest to discover the structure of DNA.
To me, there was only one way I could be happy, or 2 ways, solve DNA or get a girlfriend. I didn't get a girlfriend, so it was solved DNA.
The year ended with Watson and Crick thinking about DNA, Franklin taking pictures of DNA, Wilkins avoiding Franklin, and Pauling a distant but worrisome presence. Then, in January 1953, everything changed. News came that Pauling was indeed preparing a paper on the structure of DNA. Watson secured a copy of the manuscript and found, to his great relief, that Pauling was proposing a triple helix. It was very similar to the one that he and Crick had been shamed into abandoning the previous year. Relieved, he headed to London to share the news that the race for DNA wasn't over, only to find that Rosalind Franklin wasn't particularly interested in what he had to say.
Following his departure from Rosalind Franklin's room, he encountered Wilkins, and Wilkins took him into his room and then took out of a drawer a picture of which had been taken by Rosalind Franklin.
That picture would become one of the most famous images in all biology, Franklin's Photo 51. Jim Watson recognized the diffraction pattern immediately. It was a helix, and based on this, Watson thought it might have just 2 chains, a double helix. About the same time, Francis Crick was shown a report on Franklin's work that included an observation on the symmetry of DNA. This led Crick to a crucial insight that Franklin had missed. The 2 backbones had to run in opposite directions. That led him to the conclusion that the sugar phosphate backbones had to be on the outside, with the bases inside. So Watson started to build models again. He experimented with pairing like with like, adenine with adenine, thymine with thymine, and so on. That would make each chain identical. Watson thought that could explain how genetic information is stored. He thought he had the solution. But then, a Cambridge colleague told him that the bases could not pair with themselves in that way, and Crick pointed out that the model didn't take account of something else that was known about DNA. A few years earlier, another chemist interested in DNA, Owen Chargaff, had reported a puzzling fact about the molecule.
He analyzed the chemical composition of DNA in different species. And what he found is that the amount of A's, the base adenine, and the amount of base T's was always the same. And G's and C's were always the same.
But no one, including Chargaff, had figured out what those base ratios meant. With Chargaff's data in mind, Jim Watson went alone to the lab one Saturday morning and started playing with cardboard cutouts.
I began moving them around And I wanted an arrangement, you know, where I had a big and a small molecule. And so, how did you do it? Somehow, you had to form link bonds. So, here's A, and here's T, and I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Whew. So then I went to the pair and wanted this nitrogen to point to this one. I went like this. Whoa, they look the same. You can put one right on top of the other.
Magic.
Even if we go up to the ceiling, we're building a tiny fraction of the molecule. Hundreds of millions of these base pairs in one molecule, all fitting into this wonderful symmetry which we saw, you know, the morning of February 28th, 1953.
The model fit the measurements, both from the X-ray diffraction pictures and from Chargas data. But most important of all, the arrangement of the bases immediately revealed how DNA works.
The key aspect of the structure was the complementary nature of the bases. If you had a big one on this side, you had to have a particular small one on this side, or vice versa, and so on, all the way up. So it meant that you could easily make, by separating the two chains, you could then easily make a new complementary copy by just obeying these pairing rules of which one went with what. And that solved in one blow the whole idea of how you replicate a gene.
The structure immediately revealed two things: how genetic information is stored and how changes, or mutations, happen. The information is stored by the sequence of the bases. Mutations occur when the sequence is changed.
It's a simpler and better answer than we ever dared hope.
I remember an occasion when Jim gave a talk. It's true they gave him one or two drinks before dinner. It was rather a short talk because all he could say at the end was, well, you see, he's so pretty. He's so pretty.
I think everyone just took joy in it because the field needed it. But on the other hand, you know, the biochemistry department didn't invite us to give a seminar on it.
When the structure of the double helix was revealed, most biologists instantly recognized the power of the explanation before them. It was this beautiful molecule that could explain both the stability of life over huge amounts of time and its mutability in evolution.
Their triumph was reported in the journal Nature. It made headlines around the world and was celebrated 9 years later with a Nobel Prize.
That's kind of what every scientist dreams about, to make a discovery that has this kind of impact.
For biologists, the discovery of the double helix opened up a whole new world. It was a passport to all the mysteries of life, mysteries that biologists have been decoding ever since.
So, did you learn something? Actually, Watson died last year. You know that? Oh, yeah. Okay. Have you, lahat naman kayo dumaan ng Bio 30? So, you have watched Rosalind Franklin, The Unsung Hero?
Yes.
So, yun. So, I will not ask you to watch it. I assume you already watched that sa Bio 30. So, that is all for this week. So, we're done with in chapter 1. Can you please pass the ano ba yung next one? Okay, wala na. Please pass the
Over ilan po?
Hindi, ako na muna. Ako na mag-aano ng buong timeline. Wow. Uy!
Ilagay ko na agad yung start sa
Tama? Grabe talaga ito competitive. Daniel. Sige.
Sila.
Actually, you can spell it in different ways nga.
Alin po?
Fredricus tsaka Fredricus.
Oh, Fredrich.
Please pass the attendance sheets. Yung hindi pa nakasulat. Yung one by one, please also pass. Next meeting, ano yung ating coverage? That will be, we will have a quiz next meeting about lang dito. Very short lang na quiz. Next meeting, that would be structure and properties of informational biomolecules. We'll start with nucleic acids. This is a sort of review from your Bio-30 course, but we'll go in depth with the structure. Okay, please pass your one by one with your name and seat number. Ano?